Good morning, everyone. My name is Sophia George. I'm at the University of Miami Miller School of Medicine and Sylvester Comprehensive Cancer Center. It's my pleasure to welcome you today, to encourage you to keep participating throughout the day. The schedule for the day is up here. I've been told that our theme is identifying cell function, defining it, defining different cell types and utilizing tools to guide us, or development of tools to guide us. So it's also my pleasure to invite to the stage Dr. Nir Yosef. He is at the Weizmann Institute in Israel, and he's our keynote this morning. Thank you.
Thank you very much. Am I on? Okay. Thank you very much. So it's an honor to be here to open the third day. I was always a fan of the Human Cell Atlas meeting, so it's fun to be here and share with you some of our thoughts about where we are going with the HCA efforts and where it can lead us. So if we look at the mission statement of the Human Cell Atlas, the way it was in the first meeting all these years ago, it looks like a long time ago, it was really about the first part of the sentence here is to annotate cell types, right? To have a good map of cell types in the human body. So from the computational perspective, if you think about it, it was really all about the annotation problem. And we've been talking a lot about the annotation problem in previous HCA meetings. That was a big thing, still is a big thing. But then if we think about the second part of the sentence here, right? So to identify all cell types in the human body, it's the foundation for medical applications. So that opens the next thing, the next phase of what we can do with the Human Cell Atlas, in a sense, starting to use the milieu and the information, the data and the knowledge that we have about cell types in the human body, also to see what is wrong and what is changing in various pathologies. Now, why is this difficult? So many of us in this room can imagine all sorts of reasons why this can be difficult. Of course, there is a lot of technical variation in single-cell data. We all know about it a lot. The studies are different, batch effects, sparsity, and so on. We also have a lot of different cell subsets. It's not like we are comparing one bulk sample to another bulk sample, right? So not all the cell subsets in my data set are relevant to the pathology of interest. And of course, there is the human factor. Humans are different from each other. And indeed, even if we look at large studies, for example, COVID studies that look at the blood, we can see that the consistency between studies, just because of variation between humans, really depends on how many subjects are in those studies. And the more subjects that we have in those studies, the better is the reproducibility between studies. That means that the better is the signal that we are getting when looking at single-cell genomics for these pathology analysis. And so this has been understood by the community at large, and we are really seeing, we were seeing here in this meeting, we heard a lot about that, about increasing efforts to collect large cohorts, either across different studies like the Human Lung Cell Atlas, or in a single study like some of the CZI seed networks, right? And putting all of this together, increasing the scale and reaching to the new era, if you will, of single-cell genomics, where we don't have few samples with many cells, but we have many samples with many cells. Now, when we think about this new era, from our perspective as a computationally oriented lab, there are a lot of challenges that come in here, and I want to share with you our thoughts about some of these challenges here today. So the setup that we have is that we have a multi-donor, multi-subject study that includes a certain phenotypic variation that we are interested at. And we're going to be more concrete about it in a little bit, right? So what are the problems that we need to face with? So first of all, we have let's call them the housekeeping problems, right? The ones that I mentioned in the beginning, which is just how to take all this data and to integrate it. And how to take all this data and to annotate the cells in a way that will be coherent, that will be consistent with other studies, and that will apply equally well to all the subjects and all the samples in our data. There are also questions of sharing. How do you share this data online and facilitate that to empower the community to actually do more than what you as analysts can do on this data, but make it available to everybody, and I'm going to talk about that as well. But then this is what I want to focus on the most, which is the sample level analysis. So once again, now we don't have, we are not in the universe, so we have only many cells, but you also have many samples. So we can start asking questions about sample level analysis. So the first set of questions are differences between pre-specified groups of human subjects. For example, just case control is the easiest, but of course we have things that are more complex study designs that we can imagine. Then there is another question of de novo stratification of samples into groups, right? Without knowing anything, without assuming anything about the phenotypes, just based on the molecular and cellular profiles that we're getting from single-cell genomics, how do we stratify them into groups? And this is really enabling precision medicine. So when we think about all of those, that seems like a one class of problem, but there is another thing that we can do when we have a lot of data, which people have been discussing quite a lot, which is how to get deeper insight by generalizing to the unseen. Right, so we talked about these predictive models, some of the foundation models, some other flavors. And what I want to convince you today, or just share with you today, is that our perception is that using this kind of modeling, predictive modeling, out-of-sample predictions can actually empower quite a lot the way that we can think about sample level analysis and the way that we can think about integration. And to start with that, I want to focus on one problem that maybe has not received enough attention, but it's a very important problem of how to de novo stratify a sample population that we have based on single-cell genomics. Now, why is this important? To think why this is important, it's good to go back maybe 15 or no, to the previous decade, let's put it this way, to efforts in the TCGA. I think this is a really good example for us in this gigantic resource where we have over 10,000 samples from human tumors with various technologies, not only RNA, but also looking at the genomes and so on. And one of the things that came out from this analysis is really the power of this de novo stratification based on molecular profile of samples. And this is just a quote from one of the papers that said we estimated at least 1 in 10 cancer patients might be classified differently using a molecular taxonomy like they have in the TCGA rather than the current histopathology-based classification, right? That means that data, molecular, cellular data, like we have in single-cell genomics, can be really important in how we stratify human subjects and really enabling precision medicine. So now the question is how to divide the cohort of human subjects into groups with similar cellular molecular phenotypes in single-cell genomics. Right, so what we see a lot is, okay, you can take the data, you can divide it into different cell types, and then for every sample you can basically tally how many cells from this subject are in that type, have these frequency vectors, and compare them to each other. And that works pretty well, but there are caveats here. So the first one is which resolution is right? Which resolution of cell types we can work with? How to account for uncertainty in our data and in our definition of cell subsets? And maybe another very important point is where to look, because different cell subsets, for example, T cells versus monocytes, can induce different partitions of the patient population. So how do we do that in a more principled manner? Right, so here I'm going to pause, and now let's take a step back and think about those models that make predictions that people have been discussing here. And generally, we can think about this family of models that are generative models. What are generative models? As many of you here know, the idea of a generative model is a model that can give you some approximation of the probability distribution of the data. And based on that, through the mechanism inside the model, it can generate new observations under new conditions, right? It can make predictions. So there are various families and types of models like this, and 3 popular ones that have been used in single-cell genomics are generative adversarial, it's not generic, generative adversarial networks, or GANs, variational autoencoders, and of course, transformer networks that has been discussed here quite a lot. The large language models, those are extremely powerful. I have to say it personally, I'm very excited about those, especially after this meeting. I think there is a lot of potential there. But in what I'm going to show you next, we really focus on the tried and tested variational autoencoders that require much less information, much less data in order to give us tangible results. So one good thing about variational autoencoders is that they provide very explicit probabilistic model for the data. And you can think about variational autoencoder as a beast that gives me 2 things. The first thing it can give me, it can give me 2 probability distributions. The first one is the probability distribution of cell states. So for every cell, it tells me what is the state of this cell as a vector in low dimension, together with estimation of uncertainty around this state. The second probability distribution is around the gene expression or any modality that I'm measuring, and again, giving me an idea of what is the normalized expression level of a given gene with some estimation of the uncertainty around this normalized level. And we've leveraged this paradigm, if you will, of variational autoencoders in SCVI tools quite a lot with many different variations of this technology and many different architectures of the VAEs to answer all sorts of questions in single-cell genomics, not to answer, but to enable studies of all sorts of questions in single-cell genomics, like integration, cell-state annotation, hypothesis testing, multimodal analysis. Roman Lopez mentioned a lot of these yesterday, and I hope it's clear. And of course, the SCVI tool is online for everybody to use as open source. But VAEs can also enable us to do these in silico perturbations, right? So what's the idea here? So imagine that I have an untreated sample, let's say, just cells in resting conditions. So I can use these variational autoencoders to encode them and get a representation of cell state in the unperturbed condition. Then we can take another example that is treated, let's say with a certain chemical perturbation, and then I'm getting another representation of cell states under the perturbed state. And the idea is that we can learn a mapping in this low-dimensional embedding between the unperturbed and perturbed state. And again, we heard about it yesterday, wonderful talks exactly about this concept. And this was used in the context of VAEs to predict the outcome of perturbations. And this is an example for one study from Fabian's lab by Molot Falahi, where they used this similar idea, right, to predict the effect of chemical perturbations. But this work and many other works that have been done make these predictions in a manner that is fixed for every cell type. So when we start thinking about this problem, we start thinking about it from the let's say from the methodology perspective, and we said, well, we would like to do that thing, but we want to increase resolution. We want to make these predictions not in the level of a cell type, but at the most refined resolution that we can, and we also want to account for the uncertainty in exactly what we want to quantify the uncertainty in exactly where the perturbation is going to go. But maybe more importantly, what we thought is how do we tailor this framework, again, to go back to the question, to this very fundamental question in single-cell genomics that is going to become more and more critical as we assemble more and more data, which again is how to divide the cohort of human subjects into groups that are meaningful, that have similar cellular and molecular phenotypes. So to do that, we have this new tool we call MRVI. It stands for multi-sample variational inference. And that was the work of three fantastic student postdocs from my group, and also in collaboration with Elham Azizi from Colombia. So Pierre, Justin, and Jean. Justin now is in Colombia. So let's talk about MRVI here first. So the idea in MRVI is start with the variational autoencoder. It's the same architecture as the variational autoencoder that you know. We have these two distributions, one over the cell state, the other one is over the gene expression data. So in a sense, if you look at the first encoding, what it gives me an embedding of cells that ideally reflects only the cell state, and that's it. It's independent of the covariate, it's independent of which donor or which human subject this sample came from. It's supposed to control for everything. But now we added something else here. We added another layer of latent variables, which gives me a third probability distribution, which is the cell state, but this time conditioned not only on the data, but also on the subject ID. So in a sense, it gives me a second embedding that reflects the cell state, but also the variation among human donors. And we have the mapping between embedding that reflects only the cell state and embedding that reflects also the human subject. So when we think about the integration problem, right? This goes to something that I think Aviv mentioned yesterday, which we have these multi-donor studies. So we have this human factor, right? We have samples from many individuals. So what do? Do we eliminate the human variation, or do we not eliminate the human variation? So sometimes we need to eliminate it, sometimes we don't. And the idea here with MRVI is that it helps us hold the stick, if you will, on both ends. We have these two embeddings. Again, one that reflects only the cell state, the second one that reflects the cell state and the subject ID, and the mapping between them. It's a nonlinear function that operates in this low-dimensional embedding space that tells us how does the variation in any given cell state depends on the choice of human subject. It gives us this mapping of the donor effect. So it's good for integration. We're using it a lot now. It works quite well. I won't bore you with the details and the benchmarking, but let's now go back to the question that we started with. And now, how do we use this idea to think about stratification? So the idea is fairly clear here. Let's say that I have a cell, one cell that came from subject X, okay? And I can compute its coordinate in the sample-independent cell state space, the first latent space that I have. Then I can add my transformation that add the sample effect to get coordinates in sample-dependent cell state space per subject X. But then I can ask the question, this is where the counterfactual comes in. I can ask, what if this cell came from subject Y? What would be its coordinate in this embedding that reflects variation amongst individuals? Or what if this cell came from subject Z? Once again, I'm getting slightly different positioning for that cell in this embedding that also supposed to reflect not only the type of the cell, but also the ID of the subject, of the donor, of the human subject. So when I take all of these embeddings now and compare them to each other, what we can actually do is we can calculate a sample similarity matrix around every cell. So in a very high resolution, we can say for this particular cell state, how does my cohort, how does it stratify? So of course, this is for a single cell, but we can then average on it and scale it up to get some coherent pictures. So we have validated this approach again quite a lot, compared it to the let's say to the baseline way of computing frequencies, and based on that, calculating sample similarity matrices. But again, I won't bore you with these benchmarks. I just want to give you one snapshot of one analysis where this thing came handy. And we're going back to one of the COVID data sets. This is from Mads Hanifal's lab, where we have 130 human subjects that include both COVID positive individuals and also healthy controls. And these are blood samples sequenced with single-cell RNA sequencing, focusing on immune cells. So we take this data, this huge matrix of huge cohort, and put it through MRVI, and we're getting this embedding, the first embedding, the embedding that reflects only cell state irrespective of the donor. Then for every cell, we add the transformation. We see what is the effect of the subject ID, what would that be for that cell? And then we can estimate the sample similarities around every cell. So if you will, 130 by 130 matrix, right, around every cell that tells us what is the similarity between the cohort in that very, very particular cell state. Then when we do that, every cell, you can think about it as a vector in this 130 times 130 space, right? And then we can take these and cluster them based on that, right? We can identify cell groups that confer similar groupings of individuals. So when we do that, in this case we did principal component analysis, we find that the monocytes, for instance, they confer very similar similarity structure among humans in this cohort. So we want to see what was this similarity structure. We take the average of it, and we're getting this map right here. And note that there was no metadata included in this analysis. So the first thing that we see is that, okay, we have this donor stratification per the monocytes, differentiate between the healthy individuals and COVID. Nothing new here. This is clear. People have seen it a lot. But it's a very good positive control. But then we also see that there is another stratification inside the COVID cohort. And when we look at it more closely, we see that it separates the human subjects, the sick, the ones that are COVID positive, it separates the ones that have that just recently are recently infected from the ones that have been at least a month or more since the onset of the disease. And looking at differential expression between the different groups, it makes a lot of sense. We see lower HLA, for example, in the patients compared to the healthy controls. But then we see higher inflammatory cytokines after a longer period where we have the disease, and vice versa, we see higher interferon response in the recently infected, which again jives with what we know about this disease, but this is something that is picked up by the algorithm. And just to show you, I made the point that if I want to stratify a population of patients, where should I look? Different subsets of cells can confer different similarities. And this is the case here. What happens if you look at the CD4 positive T cells, and now we use those to stratify the population? So what we're getting in this map right here, it separates quite well the healthy cohort from the COVID cohort, but we don't get this inner stratification inside the COVID cohort that really has to do with innate immunity more than anything else, so it makes sense that we don't see it when we look at T cells. And when we look at differential expression between these two subsets of individuals, between the COVID positive and the healthy, we see again things that make a lot of sense, interferon response right in the COVID positive versus more quiescent homeostatic type of these T cells in the healthy individuals as you would expect. Right, so we saw one example here about the sample level analysis in this problem of, okay, how do I de novo stratify the human cohort into groups that have similar phenotypes? But there are many other questions here. So now I want to take a step back and tell you about how we approach these other areas, right, both on the sample level analysis, also in sharing and so on, in the context of an actual Human Cell Atlas project that we have been involved in. So it's a methodical pause. Let's not think about algorithms for a second and think about this Human Cell Atlas. So this is what I'm about to tell you is a result of one of these CZI-funded seed network projects that we have been coordinating, and it's been a fantastic collaboration. I will talk about the other participating groups in a second, but let's start with the goal. A very big question. What are the major determinants of variation in the composition and function of the human immune system? That's a huge question, right? But when we start to think about this question, what is the variation in the human immune system? So first of all, as many of you in the audience know, there are many different immune subsets, right? There are the innate immunity that is really responsible mostly about sensing and about immediate response, non-specific response. And then there is the adaptive component that is really about developing immune memory and having an antigen-specific response. We have the T cells and the B cells. There is also another important component of variation in immunity is the tissue, the position. So we know that there are a lot of studies that look at the blood and look at the circulation and look at the immune cells there, and they are fantastic studies and very informative and tell us a lot about variation in immunity, but we need to remember that the vast majority of immune cells are not in the blood, they're actually in the tissue. And when you have an immune cell in the tissue, it develops different phenotypes that have to do with interaction and with the pressures that you're getting inside the tissue, metabolic stresses, and interactions with neighboring cells. So cells, immune cells like macrophages and Tregs, for example, that exist in the tissue, we'll have different gene expression programs, again, on the metabolic level, on secretion of cytokines, and presentation of receptors and so on. Right, so that's very important, right? So are you in the lymphoid tissue or in the mucosal tissue, or are you in circulation? That's a very important determining factor. But then there is also the human component, right? Our immune system is different, right? If we compare you and I and so on. And one very important axis of variation is age. And this is something we know for many, many years. And on the bottom right here, you have one example why age is important, right? So this is susceptibility for infection as a function of age, which is the x-axis. The y-axis is susceptibility. And what we are seeing here is that, that's the dotted line, that's susceptibility, right? And what we're seeing here is that usually for infections in very young infants, they're very sensitive for infection. Then the sensitivity goes down, it stays down as we are, in our midlife, we have very strong, potent immune system, but then things get worse again as we start getting very old. And what's the idea here? Here's the hypothesis is that this is really have to do with adaptive immunity, where very young children still are not immune experienced enough to have the proper antibodies or the proper T cell cohorts, but then our immune system gets educated and responds well throughout our life, but then in old age, the adaptive immunity starts to malfunction, so we have this T cell exhaustion and dysfunction and so on that happen late in life. So this is about adaptive immunity. But then there was this very interesting humanity level experiment, if you will, that happened just a few years ago, which is the COVID infection, which is an infection that is de novo antigen, something that we have not seen, right? So the education of the immune system was not really a part of it. And here we saw something very curious, is that very young individuals were protected or were much less susceptible to the disease. And here the hypothesis is, again, this is still active research, but I can tell you just one thought here is that this has had to do with innate immunity that is more potent in those young individuals, right? So that means it's complicated, right? What is the effect of aging on the immunity? It's complicated. It can be good. It can be bad. It can be both. It depends where you look. So. So that's what we want to understand here in this project. So again, if you will, we have this three-dimensional way that we can think about immunity, the immune subsets, the tissue environment, and the age. So looking across all the immune subsets, this is really where single-cell genomics shines. Right? In a single sample, we get to have a pretty good idea of the diversity of immune cells in that specific sample. But if you think about the studies that happened so far in single-cell genomics, so at least when we started this project, right? So there are studies that you have many tissues, but few human donors, right? So then don't give us really a good idea about diversity on the age axis. There are some studies, some cohorts that have maybe one or few tissues, but for many human donors. So those can be very informative about aging, but not about the tissue component. And what we wanted to do when we submitted this proposal to CZI all these years ago was to see if we can get this more complete view of the cellular and transcriptional diversity of the immune system across tissues and the adult lifespan. So this is the team. Again, a fantastic team. This is a collaboration with Peter Sims, Donna Farber, Joe Jones, and Sarah Teichmann, and together with a fantastic group of postdocs. This has been a journey of 4 years that I'm going to give you a few insights, a few tidbits, if you will, from the results that we have as a result of this effort. So all of this work is based on two organ donation centers, one in New York and one in Cambridge, UK. And the idea was that when you have an incoming organ donation, the surgeons would try to pick tissues, and as many tissues as they can, if you will, from every human donor, usually between 8 to 12 tissues, covering both mucosal tissues, lymphoid tissues, the liver, skin, and so on and so forth, really as much as we can. And this resulted in this very large collection. We have 24 organ donors that span the range of ages between 20 and 75, more or less 50-50 between males and females. We have about 14 tissues, and all of these are about 500 samples, all of them sequenced with CySeq, with T-cell, B-cell receptor sequencing as well, about 1.2 million immune cells altogether. You can see it in this table right here. This is one of those slides that in one slide summarizes a tremendous effort that took years and a lot of money and a lot of human hours, but we don't have a lot of time, so I wouldn't dwell on it too much. So now with this resource that we have, we can now go back and ask our questions. So the first thing that we want to do is to have an integrated and well annotated as much as we can atlas of immune cells across tissues. That's the first. The second set of questions is how do we quantify the aging effects on different immune subsets and their dependence on the tissue context. Right, so if you think about these donor cohorts and everything that I talked about at the beginning, this is not a large cohort. There is not much room to do sample stratification here, but it is a complex cohort in terms of not only the human subject, but also the tissue conditions, right? So we have a lot of samples here, and it was definitely a good test case for us to think about how do we do computation for these large cohorts. So just to show you the data, so we have, again, about 500 samples from these 24 human donors from these 14 different tissues. And here we use as nuisance covariates, we think the ones that we want to eliminate, the sex, the biological sex, the organ donation center, the CMV status. We had a few other metadata, but those are the primary ones that we use. Ethnicity we couldn't use because it was too specific, but these were the primary ones. We give it to MRVI. right? So remember, MRVI gives us two latent embeddings. One that reflects only the cell states and controlling for these nuisance covariates, and the second one that also controls for the nuisance covariates, but also gives you the donor effect, right? So this is the one here that reflects only the cell state, ideally, right, and has a minimal effect of the subject ID. So that's the beast that we have. This is 1.2 million cells from all these tissues, from all these individuals. So now when we look at this data, we see that the various metadata that we have, again, like are you infected with CMV, how old are you, did you come from the UK or from the United States, male or female, and so on, those are quite well mixed. As you know, not entirely, right, because the cell state still depends on, you know, whether or not you have a CMV. So, but to the extent that we thought it made sense, we have this mixing between cell states. And we can see that we also have the major compartments, right, of innate and adaptive immunity that we can really recognize in the data, even some progenitor cells in it. So the next stage, again, this is one of those slides, like before, that we are summarizing in one slide, a tremendous effort that took, in this case, about 2 years, which is how to annotate the atlas. And as a computational group, somebody who developed some algorithms for annotation. I'm not ashamed to say that the annotation problem cannot be automated all the way. You have to have the human in the middle, and that's what we've done here. So we had this back and forth, if you will, between manual curation and refinement of annotation and automated algorithms, in this case, Cell Typist and Mimochi, which is a new algorithm that is based mostly on the protein, surface protein expression, so in a sense kind of imitating fact sorting or gating to call cell types, right? So we had this back and forth between refinement and algorithms, and we ended up with these two levels of annotation. In the first one, we think about the semi-annotated that has the human in the loop, but also the algorithms. In this case, we have about 30 subtypes of cells, of immune cells throughout. We have those throughout, you know, in the T cells, B cells, innate ILCs, and monocytes and macrophages on the right. We are continuously improving the annotation by manual curation, really looking very deeply as much as we can, right, on specific tissue sites and specific compartments and deepening it. And so far we are up to 79 subtypes throughout the atlas. We are making all of this available for annotation, so other people can actually use that in the specific tissue using that level of reference. And we saw it yesterday very convincingly that the tissue context matters for annotation, right? So that's the idea here. We make it available with POP-V, which is now integrated in the CAP platform, and hopefully this will be available in the not so distant future after, of course, we send the paper out. So yeah, I talked about POP-V previously, even in the HCA, so I'm not going to dwell on it, but the idea is that instead of one annotation algorithm, it has 8 of them, so you're getting some majority voting and try to mitigate the effect of the individual algorithm. So now the big question is, what influences gene expression and cell type composition? So the idea was to look at every compartment separately, let's say here in the B cells, and look at every gene and ask how much of the variance in this gene can be explained by the age, or by the biological sex, or by the tissue, or by whether or not you were a smoker, and so on and so forth. So when we do this analysis, we find very clearly that the tissue environment is the primary source of variation for these gene expression changes and protein expression changes in this human cohort that we have. And this is not only in the B cell, it's across all the cell types. And it makes sense because this is really about composition. For example, we see that naive and memory T and B cells are enriched in lymphoid organs. And tissue-resident T cells are enriched in the gut. And classical monocytes are enriched in the blood and bone marrow, and so on and so forth. So that really, these compositional differences are what explain a lot of the variance, and that's why tissue is so important. But then, what about the effect of age? Now, how can we learn this? So here we looked at it in many ways, but just the two primary ones would be to model the expression of a given gene with a model that takes the age, but also takes the tissue effect, the biological sex, CMV, and other technical covariates. And we use it at the level of individual genes, but also at the level of gene programs that we're able to find with the SCHPF from Peter and Donna's lab. So when we do this analysis, we find some things, some surprising, some not. So for example, in the CD8 T cells, we find that there is, in the aged population, there is a remodeled memory compartment, really this celebrated granzyme K, mesodermal PD-1 tox positive population of cells that are these terminally differentiated memory cells appear also in our cohort quite strongly, but not in one tissue, across many different tissues, lymphoid tissues, and mucosal tissues. We also see increase in inflammatory cytokines and chemokines, again, as a more universal signature of aging in CD8 positive T cells. In B cells, we saw something interesting. We saw that there is a decrease in RAS signaling in the aging population. And that was interesting because RAS is not really, the effect on aging is not really, has not been associated with B cells. It was more associated with its role in insulin signaling and the effect of this dietary restriction and the effect on longevity. But now we can hypothesize why we think it actually has to do with aging in B cells. Probably has to do with the formation of memory, with the role of RAS signaling in the formation of memory in those. And this is something that was nice if you take the same finding, right, the same set of genes that have to do with RAS signaling in our data associated with aging, and then we go to another atlas that here looks at the bone marrow, we see that very nice consistency in terms of the effect of aging in that organ-specific atlas in ours. Similarly, for the macrophages, we saw something interesting here. We see, was surprising. We see a decrease in TREM2 positive population of macrophages, again, across many different tissues. And that was interesting because these TREM2 positives are disease-associated and are associated with enhanced phagocytic activity and pro-inflammatory function, right? So, and you see it in the young individuals. So that was, we scratched our head a little bit. This is still something we want to look at, but the hypothesis is that it suggests an enhanced anti-inflammatory, but maybe pro-repair function in younger individuals. And once again, if we take this signature and go to the human lung cell atlas, and we look at macrophages there, the human lung cell atlas has many individuals there. Some of them are young, some of them are old. We look at the consistency, we find a similar signature there. Finally, if we look at the really universal signature of aging, something that appears across different compartments of immunity in different tissues, we see these themes that come up, right? As stress response that goes up in aging, apoptosis, inflammation, MHC class 2 presentation, and senescence and metabolism, like we see in the mTOR pathway that works through metabolism of senescence, and ATF3 that go through the chromatin and affect senescence. So so these are some of the stories that came out from this analysis, but now we, the analysis that I've shown you, we look at every cell type separately, all the T cells, all the B cells. I'm building one model for every compartment. But the differences that we see in gene expression can span parts of our annotated subset, or this is maybe not the right resolution, right? And subpopulations of cells that are differentially abundant, again, may not align well with how we annotate the subsets. So what can we do about it? Well, we can refine the annotation. Instead of looking at all the T cells together, we can look at subtypes of T cells and do a comparative analysis. But then the same problems still exist. So we can have even smaller chunks. But at some point, we're going to run into uncertainty. There is inherent uncertainty, and we know that as developing tools like SCVI, there is inherent uncertainty in our determination of cell state neighborhoods. Those are fuzzy. There is uncertainty there. So how do we do that? So ideally, imagine that I can take a cell, and I can ask, how likely is a cell of this state to be found in individuals of a certain age? If I was able to do that, I could do estimation of differential composition around every cell. I can also ask a composition that means like different cell types, right? Different age groups. I can also ask, how does the change in age group affect gene expression in cells of this state? Right? And that can give me estimation of differential expression, again, around every cell. So this is very, very refined phenotyping or comparative analysis that really goes to the highest level of resolution. So of course, these are mighty endeavors. It's not easy to do, but here the idea is to rely on MarVI, and this is more of an experimental part. When we thought about MarVI, we thought about it for sample stratification. But we realized we can really leverage the fact that it is a generative model to start tackling these questions. So the first question we can tackle by going from the embedding the reflex only cell state to the embedding that as a donor or the subject ID effect. And similarly, we can look at differential expression because we can decode this representation and get counterfactuals or expected, posterior expected gene expression profiles given a subject ID. So when we do this analysis, we are able to find things that are really at high resolution. So for example, if you look at the CD8 T cells, we can find per cell, per gene, right, what would be the estimated effect of aging. Then we can cluster the genes for genes that have similar effects across the cells. And that gives us all these gene modules, like this granzyme K positive, terminally differentiated effector memory gene module that I mentioned before. But now we can actually locate it in this UMAP of CD8 positive T cells. And similarly, we can do this differential abundance at this high resolution, and we find in this case this transition from a naive state, right, to a less naive state in that compartment. Not surprisingly, in the monocytes, in the macrophages, we find something that is a bit newer and unexpected, which is this population of pro-inflammatory gene expression program that increases with age, but only in a subset of cells. Right, so just to summarize this part, the idea here is that MARVI is this approach of adding the donor effect explicitly to our variational autoencoders, give us a way to do better integration, because again, we can hold the stick on both ends, but also to do sample level analysis at this arbitrary level of resolution, if you will, right? And we can do de novo grouping of cells based on sample level properties, like I've shown before. We can use that to find, we don't need to start with a priori definition of cell subsets. We can actually let the data decide for us how to subset the cells based on the phenotype, based on the effect, rather than based on what we perceive to be their label, right? So it can give us similarity between samples, overrepresentation of a given sample group, or differential expression. And I have to say that the bottom two are not an entirely new concept. We have to remember that there was some work here from John Marioni on Milo and Smita Krishnaswamy as well. But here the idea is to take it to the next level in a sense and really think about uncertainty, quantify it, and do it all in the same model. So not to separate the dimensionality reduction, if you will, from the comparative analysis. So the last thing that I very quickly want to show you, almost out of time, is now that we have these big atlases and so on, so how do we share online our results, but not just to share the data, but to share something that you can do something with. So we saw some of this yesterday with SCGPT, which I think is fantastic. So here it kind of goes in the same direction, right? So what else can this atlas do for me and how? So when we have the atlas data, I can use it to interpret and boost the use of my data, not only for annotation, but also to increase power and to do reference-guided dimensionality reduction, deconvolution of spatial data, and so on. So the way that we usually do that, we download the data, train the model, and then do something. And the idea was to see if we can cancel the download the data and train the model and go straight to the do something part and really facilitate a quick use of this kind of models that we are presenting. And for that, we have this new project. This is a CZI Data Insight funded together with Fabian's lab. We call it SCVI Hub, which is a platform for sharing and reusing models, and it is all based on the Hugging Face. For the AI people here will probably recognize that name. It's a platform for sharing of AI models that is very version controlled and it works very well, very nice API. So we have all these models there. So far we loaded over 90 data sets as models to Hugging Face. And what you can do is you can download the model and directly pipe it to many types of analysis in ScanPy, SCVI tools, and other packages on SCVerse. And one interesting point here is that you can download only the information that you need, right? So that really saves you, especially if you are low on resources, on memory resources, for example. So if you want to do an atlas-guided embedding, you just want to project your data on the low-dimensional space and look at it, or you want to annotate it with ScanVI, you only need to download the model. So that's a small fraction of the size of the data. It requires much less bandwidth, much less resources, both compute and memory. If you want to annotate with other methods or do deconvolution of spatial transcriptomics or use the decoded expression in the atlas, you only need to download the cell state embedding, which again is a compressed representation. Instead of every cell being a vector of size 5,000, every cell now is a vector of size 20. And you can actually use those decoded values to do differential expression, to actually look at the expression data, even if you don't have it, because you can actually decode it through the VAE, and we've shown that it works quite well for differential expression. And of course, you can download the raw counts if you want to do things that are more complicated, like revise the model or augment it with additional steps. Right, okay, so that's my summary. I've shown you these two components here. The first part is how we think about these large cohorts and all the challenges that I think are there, and there are a lot of outstanding challenges, and I'm very excited to see what comes up from people here and generally from the community, and thinking about how do we make more of human cohorts. And I told you about our endeavor into a human cohort and trying to look at immunity from in this three-dimensional perspective of the tissue environment, the immune compartments, and the age. So so I already acknowledged our fantastic immune aging network, all the participants, the postdocs and the colleagues that were part of it, also the 2 organ donation centers and the surgeons that were part of it. I want to thank my group, the funding sources, and of course for the donors and their families. Thank you very much.
Good. Shin?
Great talk, Nir. My first question is, can you join the Genetic Diversity Network?
Absolutely. Yeah, we have a lot of interest in that. For sure, we should talk.
Thanks. Second question is, if I can have one more. I was struck by how the collection site had a big effect despite regressing out or controlling for the donor. What explains that? So
there are some residual variance from the collection site that can be explained by, there is also always a tension between how much you correct and how much stays there, and if you're doing it too much and so on. But there is also, if you look at the actual metadata, it's not perfect. It's not the perfect experimental design, right? So there are, if you look at the ethnicity, there is bias there, because if you compare people that live in the UK, people live in New York, it's not exactly the same, right? So that's the residual, I guess, is both a component of the technical, but also the population of people that live in that city.
So some combination of technical effects from sample collection and then just environment and genes. Correct. And something like that. Okay, yeah. Thank you.
We have a question over here.
Hi, Nir. That was a great talk. I guess along the same lines, I was wondering, because these are organ donors, I guess, people who have recently died. So do you see disease effects in the data? And
yes.
If not, or if so, is there something interesting there that you can pull from it?
No, that's a great question. We did not look for these, but we do see epitope specificity for COVID. But the people, most of them did not die from, we have one person died from a brain tumor, but mostly without a condition that would inflict too much on the samples. We have different types of death, cardiac and brain. And these are all covariates that we try to pay attention to, but it's definitely not perfect. The numbers are small, so too small for us to say something meaningful.
Thank you.
Hi, thanks. We've got some questions from the virtual audience. 2 questions on MRVI. First from Mark Elouseur, saying, thanks, very cool talk, Nir. One question, what are your thoughts on applying MRVI to metacells, like C cells, for example, except instead of the individual level?
It's possible. I don't see why not. Once you abstract your data into metacells instead of single cells, you should be able to do that. There is a bit of it's almost 2 approaches, right? How to handle the noise, the biological noise in the data, and the sparsity, which is why we want to do metacells. We say, okay, no, single cell is not very informative. It's just a point in some cloud. Let's just take a few points and average them together. In a sense, variational autoencoders and even the foundation models that we saw, they take a different route. And through the model itself, they're trying to do that smoothing. So that might be conceptual double dipping, but in practice, I don't see why it will not work.
Thank you. And the second question was from Dana Peer. She finds MRVI really exciting, but autoencoders are not magic. And they don't know batch from real biology unless they're properly trained. So what under the hood allows it to distinguish batch from biological difference?
Yeah, no, it's just once you start having enough samples, right? In our case, we think about the batch as not as the donor, the idea of the donor, but the sample collection site. So in that case, if you have enough of them that come from every site, you can start teasing out some variation there that has to do with that. But of course, this is not magic. If I portrayed it this way, that's completely not the idea. It's definitely difficult. The models are far from perfect, and there is definitely residual signal there that is technical, and we are definitely eliminating signal that is biological. There is no doubt that that's what's happening.
Go ahead.
That was just absolutely delicious, and can't wait to get that data. So I think you a bit under-described the nature of, if you look at gene signatures as a function of cell class, subclass, cell type, and maybe substate, how different are those DEG lists per what you really believe are different levels of the class structure as a function of tissue? And so it seems to me that there's very significant differences in cell adaptation to being in tissue environment.
Yes.
1, 2, 3. And what are those genes, and to what extent are we now talking about really different functionally altered versions of what we think of as this effector CD8 T cell that has this effector set coming out of it?
Yes.
What's contextualized there?
That's a great question. I left out and maybe skimmed over this a bit too much, but I left out the tissue specificity part of this analysis, which is a very important component of it, right? And when you do this, you find these tissue signatures that can be subset specific, or it can even be universal. So we definitely found quite a few of those. So it's a huge amount of information, right? We couldn't do everything with it, but we definitely find a few of those. I'm happy to show you some of the data offline. I don't have it just fresh in my head right now, but that definitely exists, especially if you're comparing the mucosal tissues to the lymphoid tissues. That's where you see
everything.
We know that those are
class structure even to the tissue environment.
That's correct. I mean, we actually use MARVI to cluster the tissues, and we get this. What you would expect. I mean, basically mucosal and lymphoid, and then blood or blood rich.
But there's more than you would expect as well. That's I think a sort of a hidden control layer that has to do with immune cells in different environments functioning. I wonder if do we have any priors on, let's say you do start from a transplanted set of cells, and then they localize to different tissues. How fast do they adapt to the tissue environment?
Oh yeah, no. So this is so interesting, right? And I know that there are. I mean, of course, this is a problem that with a very large beard, right? And it's an important problem. But now, if you think about single-cell genomics and technologies, an exciting set of technologies are allowing us to look at this tissue adaptation at higher resolution, right? And I'm just going to quote one work from my colleague at Weizmann, from Ido Amit, who has this labeling technique that he calls MANSIC, right? And the idea is that you label cells in circulation at different time points. So basically, the label would tell you what is the last time more or less the cell was in circulation. Then you can look at them in the tumor, and then see what is the extent of adaptation that you're getting as a function of how long you've been inside the tumor, right? So these kind of technologies, I think, would really open up this in a more fundamental way.
Can I just ask one more quick question?
We are at time, and the other plenary session has to.
Really important question.
But we can ask him. You can ask him after the session. So I just want to say thank you very much. It was a really engaging session. We want to make sure there's time for others.
Thank you.
Introduce the next session, which is how should we think about cell function, and get going on that. We have 5 speakers in this one, although 2 are sharing a spot. Our first speakers are Katy Börner and David Osumi-Sutherland, and they're talking about the Human Reference Atlas and functional tissue units.
Good morning. So I hope the slides made it, yeah. So this is really not just my work. This is work across multiple NIH-funded consortia. CIFA is now also supporting the multi-scale human. You might have seen that. And it's thanks to the organ networks that HCA has created and many different consortia that this work exists. So on the next slide, you see more information on functional tissue units. On the next slide, I have to do that. I can do that. Sorry, I'm used to Zoom meetings where somebody else runs his slides. So we have our own definition. It took a while, but we define tissue units as the smallest tissue organization that performs a unique physiologic function, and that is typically replicated many, many times in an organ. So for instance, in each of your kidneys, you commonly have 1 million glomeruli if you're not born prematurely. And that's a lot of redundancy, and that's good if if disease strikes, you can actually lose some of them, but still function properly. These functional tissue units, we believe, are a really important bridge from the rather large organs down to the single cell level. I also personally believe that single cells, only a few can do a lot, but most of them actually operate as a team. And so it's really, really important for us to understand what teams exist, at what level of granularity, how do they work together, who is in the team, and how do they impact each other. It's like a tango dance, if you wish. They really have to work extensively with each other. If one gets tired, others might actually fill in. And so here you see really the enormous scale we are trying to cover when we try to map the human body across scales. And you also see the renal corpuscle here in 3D, and I think 3D really, really matters. So if you have 3D data that's available, please see me. I would like to have more 3D data. And so we have embraced looking at these functional tissue units across organs. We now have 2D renderings for 20 FTUs across 10 organs. And we get experimental data mapped to those 20 FTUs via Asimus or other cell type annotation tools that are aligned to the AST+B tables. And this way we know what cell types are commonly located inside of which functional tissue units. And we can also go the spatial route where we use code developed in Kaggle competitions to segment out functional tissue units, and then we know what cell types, single cells, are inside of these functional tissue units. And we can compare these two, of course. And we see also over time, using this interactive tissue explorer in the making, should be out very soon, definitely for Christmas. What happens as we age, what happens as disease strikes, and on the left-hand side you can select one of those 20 functional tissue units. So we hope many of you contribute data to this, but also start exploring these teams of functionally valuable cells. In follow-up work, we are now trying to connect these 20 functional tissue units to the vasculature common coordinate framework. And some of you might be familiar with this work. This is really an effort trying to get us a latitude-longitude system for the healthy human body, male and female. And you really start from the heart or here down to the kidney organ, and you go down, down, down to the microvasculature. And over the last 4 years, we have created a database of more than 1,000 vessels, also branching angles, diameters now coming in, thanks to another Kaggle competition. And there are 3 HubMap pitches going forward, which will get us data for the single cell composition in different organs and in different parts of the vessel system. So again, if you're working on blood vessel data, please let me know also. There is a Kaggle competition going on. $50,000 prize money will be given to the winners. So if you have students that would like to participate, there's a few more days left before the team merger deadline. You just go to Kaggle and you search for hacking the human vasculature. Ultimately, I believe we will see very different morphology and gene marker and other biomarker expression values across the human vasculature, and probably a combination of morphological features and gene and other biomarker expression values will be relevant for getting us an understanding of where we are in the human body. And I'm leading over to David, who will explain how to get to the function of some of those teams.
Thank you, Katy. Okay. Right, so I've really just got some thoughts there on annotating cell function. A lot of the discussion, there's a lot of focus on annotating cell type. I should introduce a little bit what I do, actually. So I build ontologies and build tools for using ontologies, so one of the main ontologies that I work on building is the cell ontology, well, one of many actually. So just very quick introduction to that. There's actually closer to 3,000 cell types in there now, rapidly growing, used by very many resources as a way of standardizing annotation of cell types. So it allows for cross-search, it allows for grouping of like cell types using its structure. One of the projects that I work with is, one of the big challenges is how we actually get broader usage of this standard rather than just simple annotation with free text. And so working with the cell annotation platform, we're building a platform that really makes it much easier to use these standard cell type terms. And also, it doesn't force this. Annotators are able to use their own annotation terms as well, and link those to the cell ontology terms. It's got this quite reasonably sophisticated autocomplete system, which is tuned to sort of boost, if you're working on a particular organ, say the kidney, it will boost cell types up that are known to be in the kidney in the return in this auto-suggest. So that's sort of the general context, but here, as this session is about function, I wanted to think a little bit more about annotating function. So here's an example, actually, from a whole mouse brain atlas that's under construction. So there's a new browsable version of this out via tools at the Allen Institute. I think that's come out today. And one of the things in this atlas, and that we see very regularly with annotation of neurons, is people want to be able to annotate neurotransmitters. So this is, rather than, so if you look at the UMAPs here, there's very fine annotation of these UMAPs by transcriptometrically defined cell type, but then if you look at, say, if you look at, for example, GABAergic neurons there, you'll see neurons on opposite sides of the UMAP annotated as GABAergic. So this is, functional annotation very often tends to be cross-cutting of transcriptometrically defined cell types. It's another case where, and there's very many types of functional annotations you might want to use. If you want to be able to standardize this, how could you go about doing that? Well, actually, in a sense, we already have a pretty large and well-curated ontology that we can use. So if we define function, I think one reasonable way to define function is the ability or capability of carrying out a particular biological process. So a GABAergic neuron is capable of secreting GABA as part of neurotransmission. So there's the process. So we have this very rich hierarchy of well-curated hierarchy of biological processes from the gene ontology. There's almost 29,000 terms in there now, very richly structured. So very many of you are familiar with that from just standard gene set enrichment, but it actually gives us a way potentially to directly annotate cell types where we've got evidence for function. And there's actually, in building these ontologies, we have thousands of curated links between cell types and Go. So here's just a few examples I harvested. So from the, and these work in both directions, between cell type and Go and between Go and cell type. So we've got links from spermatid to the Go term for acrosome assembly. You can see a link between basket cell and GABA secretion in neurotransmission, and that's actually used to automatically classify basket cells as GABAergic neurons. And then links in the other direction. So you can see a link between platelet degranulation and platelet. And then you've got the whole structure of the ontology above that that's potentially useful. So I think one thing that perhaps is potentially that we could work with here is that, there's the conventional way that most people use Go in gene set enrichment, or potentially other approaches with the gene sets that you get from Go annotation. So I can go from one of these terms, I can get a set of genes that are annotated as being involved in this particular process. So this gives us a bunch of links that we can potentially mine in order to provide evidence for cell type annotation. And actually we're working on, as well as I think potentially folding this into the way the cell annotation platform works, we're working on on a bunch of Python libraries to make that more accessible. But also, so I said, I guess a lot of people think of the gene ontology as something that is about only classifying genes. It's actually just a set of very general ontology terms. So you can just as well annotate a cell with a gene ontology term, to record its function as you can annotate a gene. And then I think there's potential here also, and this is really just to kind of get an open discussion of what the best way is to formalize this and what the interest is. You can annotate gene sets derived from single-cell RNA-seq data. So I think getting beyond this sort of atomic level of this gene is involved in this particular process, what we want to know is whether there's a whole cassette of genes, a whole module that's expressed that's sufficient for carrying out the process in order to accurately annotate. So I know there's lots of work deriving, programmatically deriving cell sets from single-cell RNA-seq data, and there's, so I'm very much interested in the potential for direct annotation there. And of course, we need practical tools for being able to do this. This is not something we're working directly on the cell annotation platform right now, but I just wanted to put it out there that if we had an ability to annotate by function and not just by a general cell type term, how much interest is there and what kind of evidence should be included in there. Anyway, I'm out of time, so there's some acknowledgements. Any questions?
It looks like we have a few minutes for questions. Are there any? There's two of them over there.
Well, why don't you force annotators to use the ontology?
It's kind of hard to force people to do stuff, I think that's, there's carrots and sticks here and nudges, and I think there's, in the context of a particular standardization effort, you can specify that people must do this in order to submit their data to something. There's plenty of precedent for that with journals, using star methods or submitting accessions for your genes or whatever. There's lots of precedent there. So I think it's a matter of having the right kind of carrots and sticks, and that depends a lot on context. So part of it is with things like the cell annotation platform, just how we make it easier.
I mean, you could use them to extend the ontology, right? Presumably they're not using the terms because they're not finding the one they want.
Well, I think, sometimes the terms are a bit long. I think any annotation process, people have got to be able to use their own terms in addition to the cell ontology terms, They want, I think it's just a fact of annotation,
Did we have a second question over there?
Malta's got a question.
Thanks a lot, David. A really interesting talk. Malta Luke and Helmholtz Munich. I've used kind of Go terms to try annotation as well with enrichment, and that sometimes worked well, and sometimes not so much because not everything is known. I think in terms of function. How do you set up these Go term cell ontology links? Is that literature curation, and how do you record the evidence for that?
Yeah,
link?
so these are actually. These are a long-term effort on the part of the ontologists. So there's very many curated by Go curators over the years. If they're curated by Go curator, there'll be a reference connected to the term which supports that as well. And the same works on the cell type side. So we can mine the ontologies for references to support these. And then when it comes to the links to genes, obviously Go annotations will come with links to evidence and references, so that's potentially mineable as well. But I think getting beyond, gene set enrichment is a very limited technique with a lot of problems. It's useful, but I think getting beyond thinking of simple gene set enrichment and thinking about more sophisticated ways, or ways that actually really expose the assumptions that we make when we do these enrichments.
Is it a super quick question?
I think we are already facing the issue with the breast where some groups use the term luminal progenitors for the same cells in a single cell setting, others use luminal secretory. So maybe getting buy-in by the top journal editors where they can enforce a specific terminology may be something to consider in future.
Yeah, and we can support both of those. And, you know, one will become the primary name, but we can support the synonyms with references as well and allow search on those, so it's not insurmountable.
Okay, let's thank David and Katy. Our next speaker is Gray Camp on integrating a human organoid atlas. Thank you.
All right, thank you. Can you hear me? Test? Is it on? Microphone? Yes. Right, yeah, I'm really excited to be here today. I'm actually representing a lot of different people to tell you about our work on towards a human organoid cell atlas. I'm currently a group leader at the Institute of Human Biology in Basel. And altogether, we really like to think about how human developmental biology and how the differentiation of diverse cell types is really orchestrated within these complex tissue environments. And it's a particularly exciting time to be studying human developmental biology because we have not only these great atlases, but new model systems to kind of recapitulate some of the developmental architectures and functionality that we see in the primary tissue. So as we all know, from any individual, you can take a differentiated cell type, reprogram it into a pluripotent stem cell. These stem cells can be used to differentiate various cell types in controlled culture environments. You can also provide a three-dimensional environment and certain cues to allow these stem cells to develop into tissues three-dimensionally, often called organoids. Another source of cells for complex human tissues in 3D are tissue-resident stem cells, either from the fetus or from the adult. And altogether, these systems really provide avenues to model health and disease. They provide a number of really interesting features as well that we think might help enhance the overall human cell atlas. So they can get access to really difficult to obtain embryonic time points. You can do time course measurements. You can do genetic environmental perturbations, in some cases high throughput screening. You can test hypotheses from the atlas in this controlled system. You can engineer the tissues to be more complex and model some aspects of inter-individual variation. Kind of led by Christoph Bach and Barbara Treutlein, we wrote a short paper on what we envisioned for the organoid cell atlas to encompass. As I mentioned, there are different sources of organoids, adult or fetal stem cells, or these pluripotent derived organoids. We can use the organoids for various biomedical applications to model aspects of organ development and physiology. They provide kind of patient-specific avatars for personalized drug testing. And in some cases, people are trying to develop these systems towards transplantation therapies. Now, we think that combining with the human cell atlas, we can really address some of the major challenges in organoid biology, providing a system to validate existing organoid models, kind of give ideas about how to develop new organoid models or to adjust their components. Generally we want to be able to apply these organoids to understand how the human genome really can regulate function. We've been working on this problem for quite some time. We've published some papers back in the SMART-Seq2, Fluidigm C1 days, studying cerebral organoids, comparing cortical regions to the primary tissue, liver organoids to the primary tissue. And what we find is that these iPS-derived organoids really can model important aspects of fetal development. And so what we wanted to do is build up a more comprehensive survey of what's out there right now, organoid single-cell RNA-seq data. We've broken this down into really two components, the brain, and we're trying to kind of acquire all endoderm-derived tissues. Right now, we really have a lot of data on gut and lung. And so with this time, I want to tell you about where we are. It's really a work in progress. It will probably always remain a work in progress, at least in the organoid space. But first focusing on the brain. The brain development is complex, as you know. In the beginning, you form these different cephalic structures, forming the telencephalon, the diencephalon, the mesencephalon. And these are really coordinated by morphogens that help orchestrate the development of these units. And within each unit, there are progenitor cells, intermediate progenitors, and neurons that develop and acquire identities for each of the major brain regions. And we're able to model this in really interesting ways in organoids. So we can take iPS cells, aggregate them, induce the neuroepithelium, and then in some cases we let the organoids really self-guide themselves, and they will form multiple brain regions. In this case, it's a cerebral organoid protocol from the Lancaster lab. It can give rise to these complex cortical architectures that have progenitor cells surrounding a lumen and then differentiated neurons that begin to layer. Of course, you can also use particular morphogens known from model systems in developmental biology to really guide the formation of different brain regions, the forebrain, the midbrain, the hindbrain, and so on. And by now, there are a lot of data sets out there covering these different brain regions. And so we've started to download them all the ones that are publicly available. We have brought together 25 publications, 36 data sets, about 1.8 million cells. We've curated everything, provided consistent preprocessing, metadata, harmonization, and then started to integrate using auto-annotation and label-aware integration. Using tools from the Tice and Yosef and other labs. So this is kind of the coverage we have right now for the organoid, the brain organoids. We span really from very early, a few days in culture, up to 450 days for all these for some of these protocols. The vast majority of the data is from 10x, but we do have some other data modalities in there. And this is what the integrated UMAP looks like. So you can see that we're covering parts of the progenitor, intermediates, and neurons from the dorsal telencephalon or the cortex, the ventral telencephalon, parts of the midbrain and hindbrain, choroid plexus, early stages of the naive neuroepithelium, and then the originating pluripotent stem cells. We also have astrocytes and oligodendrocyte progenitor cells in the atlas, and some off-target cells that come in some of the protocols, neurocrest and mesenchyme. I think as you can see that it's really nice coverage of both guided and unguided protocols. So cells in the unguided protocols intermix with cells from the guided protocols. Again, we cover really a large time series and lots of different publications. And so what's interesting is that you can start to look at the composition of cell types and brain regions in each one of these samples. And you can see that there's lots of variation between the composition depending on the organoid type, right? And this has somewhat to do with the morphogen guided protocols, but also there's like a lot of variation in the different unguided data sets out there. And so now we kind of capture this and we can start to explore really what's going on in these systems, what's driving it, how variable are the cell types. Another thing that we're starting to look at is how similar the cells are to a primary atlas. So Stan Linderson's lab has built up a really nice map of the developing fetal brain, which is shown here on the left, covering all the brain regions, a lot of the cell types and states that we see. And so what we're doing now is like projecting the organoid cell atlas onto this fetal atlas. And we're trying to figure out what have we covered so far, what are we missing, how similar are the states in the organoid compared to the primary tissue. And there's really a lot of analysis we're doing here. I just want to show you one example. If we take a look at the kind of the presence of organoid cells that are projected onto the atlas, we see that some are missing. So for example, Purkinje cells are really poorly represented in any of the organoid published data sets today. And really, there's other cell types that are missing. There's some cell types that are really well covered. And so what we're wanting to do is kind of use such a system to really guide and benchmark new protocols. And we would hope that from different organoid methods that we have out there, we really can capture most areas of the developing human brain. And right now, again, there's still some missing. So this is where we stand. We're working on nice tools to project and benchmark new protocols. And now I'm going to kind of switch gears and tell you about where we are on this endoderm-derived organoid cell atlas. So as I mentioned, we have from pluripotent stem cells, we can differentiate either to the endoderm, the mesoderm, or the ectoderm. If we look at the endoderm through induction by activin A, we can form through various morphogen compositions, stomach, intestine, lung, thyroid, and liver organoids. This is an example of intestinal tissue generated from iPS cells. And so in vitro, you can specify the intestine, and then you graft or transplant the organoids into immunocompromised mouse, and you get this really complex tissue forming. So on the top row, this is a primary tissue, 8 to 18 weeks gestation. And the bottom row is this transplanted human intestinal organoids, up to 8 weeks of development. I think what you can see is really complex epithelial architecture, subepithelial mesenchyme, smooth muscle cells, and this is all derived from human pluripotent stem cells. We can also generate mostly epithelial organoids from tissue-resident adult or fetal stem cells covering different endoderm-derived organ systems. For each one of these epithelial organoids, there's often differentiated cell types. This is for the intestine. Intestinal organoids can really cover these various absorptive and secretory lineages, right? Okay, so we've went in and tried to acquire as much data as we can find, covering adult, fetal, and iPS-derived stem cells of endoderm-derived organs. We've integrated it around 800,000 cells from 55 publications, and what we're trying to do is annotate the cell types, map to primary tissue, compare the adult, fetal, and iPS-derived organoids, and then set up a system to project new data sets so it can continue to grow. I should say we focused a lot of our analysis on lung and intestine because that's where we've been able to acquire, there's the most data published so far. Most of the data is 10x, and again, for intestine and lung, we have iPS, fetal, and adult-derived stem cells. For the other tissues, we have less coverage. So if anyone out there has any data sets that they want to contribute, let us know. This is what the UMAP looks like. Again, you can color the cells based on the tissue that they're trying to model. The vast majority of cell types that are in this atlas so far are epithelial. We have also quite a lot of mesenchyme from the iPS-derived organoids and some endothelial neuronal cells as well. We can get finer grain annotation of each one of these cell types from each one of the organoids. I won't go through the details, but this is what we're trying to understand how to name and these cell types that are deriving from the organoid. And our plan right now is to go into really a deeper analysis of the gut and lung data sets. And I'll just tell you one example of that so far. So this is the integrated, if you pull out only the intestinal cells, you can then color them by cell type, whether or not they're duodenal, ileum, colon, or pluripotent stem cell derived, whether or not they're fetal or adult. And what we can do is we can start to pull these cells out and now compare them to a reference atlas. And what we find is that if we have fetal and adult epithelial cells, what we were expecting is that the fetal organoids would map to the fetal and the adult would map to the adult. But we don't really see that. We see the adult mapping to the adult, the fetal also often mapping to the adult, but the transplanted iPS-derived organoids really map to fetal. Right? And so somehow in the culture that we have for the fetal-derived stem cells, we're getting some sort of maturation happening. So to be able to model the fetal state, we need the pluripotent stem cells currently. We don't know how or why. We're trying to look into this. Also, one thing we're looking at is kind of variation between the protocols. And it's really interesting for these iPS-derived cells. In vitro, they're very immature, and you have to transplant them in order for them to mature. But there is some variation in the protocol where we can find that certain protocols get differentiated cells. And so this is one protocol where you add NRG1, it's a mesenchyme-derived ligand, to the culture media in vitro, and you get differentiated cells. And this is different compared to other protocols, right? And so what we're trying to use is figure out like what are all these protocol variations and what cell types that they're associating with. Okay, I think I'm out of time, and I just want to thank, there's a really big collaboration between Barbara Troyline's lab, Fabian Theis's lab at Helmholtz, Barbara at ETH Zurich, and our group at IHB, and we're looking to collaborate with more people. So with that, I can stop, take questions. Thank you.
Okay, I'm told we do have time for one or two questions. There's one over here. Please state your name and where you're from.
Thank you for nice talk. I'm Yoshiaki Tanaka from University of Montreal, and yeah, it's very nice, great atlas. But I think organoid development is highly dependent on the resource of the stem cell. If stem cell quality is not good, maybe organoid differentiation also not good. So in your atlas, you also focus on the starting embryonic stem cell and starting information.
I mean, that's definitely what we want to kind of focus on. We haven't exactly teased out all the details yet, but for the brain atlas, we know that in unguided protocols, you'll get differential proportion of different brain regions. And this is dependent, we think, on the line, as you say. But it's kind of interesting. Some of the data sets we have the same line, but guided. And then you can kind of compare what the variation is between a guided protocol for the same line and unguided. So this is an example of what we're looking at.
Thank you.
Yeah. Also the age of the stem cell donors. It's interesting.
Yeah, exactly.
Okay, we can have one last question. It looks like we've got one at the back.
Great talk. I'm Jongwon from KAIST, and I have two quick questions. One is, have you remapped the data, or did you start from the matrix files for the atlas composition? And the other thing is, can protocol be quantified? Like, you have a different version of protocol and different amount of like Wnt and other ligands. Can you actually vectorize that, and can you actually put into like MRVI, like Nir Yosef has shown today?
Yeah, it's a great question. So for the brain organoid atlas, we started with the processed matrix. For the other organoid, we downloaded, for the most part, we downloaded the data, remapped it raw. But I think we want to move towards just using the processed data, certainly. For the protocols, looking at what's underlying the variation between the protocols, using these tools, Nir and others have been presenting, that's definitely where we want to go. We're trying to find a way to systematize the protocol. And this is not too easy. A lot of people that report their data don't report so clearly what they did in the development of the organoids. So we're trying to go into the publications, tease it out, put it into some sort of matrix that we can play with. Yeah, it's important.
Okay, we have to move on in the interest of time, so let's thank Gray.
Thank you.
And introduce our next speaker, Brian Brown, who's going to talk about cancer cell control of the local cell environment.
Thank you. Thanks. Let's see. Here we go. So I'll talk about our work, which is really focused on trying to understand how cells control their microenvironment. And we're particularly interested in how cancer cells do this. This is an image here of a melanoma, which is really fascinating because you see in the middle there the cancer cells in pink surrounded by a layer of macrophages, and then outside of that are the B and T cells. And this type of structure is really predictive of how the patient will respond to immunotherapy. So it becomes very, very important clinically, but it's also important from a conceptual kind of point of view of what are the genes that are orchestrating this type of structure where, no matter what happens, and this is quite fascinating, even if this melanoma metastasizes to another tissue, it still takes on that type of structure. And so what is it that does that? So my lab a few years ago developed a way of doing CRISPR screens in situ, so you can resolve them by spatial imaging. And we call it Perturb Map, play on Perturb-seq. It was published a few years ago, so I'm not going to go so much into the details. But what we do is we take a library of CRISPR vectors encoded in a lentiviral vector, and they have this protein-level barcode that we developed. We drop them onto cancer cells. In this case, we use lung cancer cells, and you can go up to 120 different CRISPR perturbations. And then you implant it into a mouse, and then you take out the tumor and you do multiplex imaging in order to resolve the different protein barcodes, and that tells you what CRISPR a particular cell is carrying and then what knockout they have. And the power of the technology is really that we can not only do CRISPR screens where we look for enrichment or de-enrichment of a guide, which predominantly focuses on cell intrinsic phenotypes, but we can also, now that we can resolve what's happening around a cell that has a particular perturbation, we can now look at what happens or what is a gene's function cell extrinsically. How is it that cell or that gene enabling a cell to control its microenvironment?
We can also look at genes with extracellular functions, which is not normally enabled, resolved in a CRISPR screen. So if you have a genome-wide CRISPR screening, you knock out a bunch of cytokines or chemokines, but 1 in 1 million cells in a Petri dish have that knockout, all the other cells are going to compensate for that. So in these genome-wide CRISPR screens, you're actually not really assaying for extracellular gene function. So this is the power really of the technology. And this is what it looks like when we injected a mouse with a library, or cells with a library of 35 different gene knockouts. This is an example of one lobe of a lung, and we get many, many different lobes, so we actually are able to resolve a lot of these. So it looks like you have one big tumor mass here, but then when you stain for the different protein barcodes, you're actually able to see that there's very distinct homotypic clones here, and each clone has a different gene knockout. And the power is that we can look at that and we can say, okay, well, now for each gene, did I get an enrichment or a de-enrichment of tumors, but also did I get more lesions? Did I get more cancer cells? Where are those cancer cells? And what is the immune microenvironment in each lesion? So this is a piece of the data where we're able to look at what happened to each gene knockout. And the thing that we found, I just want to focus because this is published data, and you're welcome to go and look at the paper, on two genes here, which are SOX1 and TGF-beta receptor 2. So when we knocked them out, we got the biggest and most frequent tumor lesions, more than we would have expected by chance and more from the input. So that was caused by knocking out those two genes, we get an enrichment of those tumors. But what was interesting is then we could stain all these cancer tissues, and we stained 1,700 different tumor lesions, and we looked at the different immune compositions of those lesions. And what these radial plots show, if there's a bar on the outside, that means there was an enrichment of that marker-positive cell. And if there's a bar on the inside, there was a de-enrichment. And if you look here, the two tumor types that grew, the two knockouts that resulted in the most frequent tumors, actually had completely opposite immune compositions. So when you knocked out SOX1, those lesions had an infiltration of T cells, CD4 and CD8. And when there was a tumor that had a TGF-beta receptor 2 knockout, you had a depletion. And we went and said, okay, well, what happens when those two are side by side? Are the immune microenvironments going to mix? Because you have this kind of very drastic difference. And what was really striking here, and I think this speaks to a lot of how biology is working, is the microenvironments remain completely separated. So this, if you look at the H&E histology, it all looks like one tumor. But if you see the cells in white there are the SOX1 knockout, and you can see on the left part of the panel, you can see that there's all those red and green dots are the T cells that have infiltrated. But almost to the cell, where you have the cancer cells that have the TGF-beta receptor 2 knockout, you can see that there's no red and green cells there, that it's remained completely immune excluded. So this completely confounded me because honestly, I would have thought the T cells just started to traffic wherever they could go. But there's something there when you have this TGF-beta receptor 2 knockout, a structure that it's creating that's keeping those T cells excluded. And this is similar to things that we see in patients. Then we went and did spatial transcriptomics using the Visium platform. And what came up in the TGF-beta receptor 2 knockout is that the most upregulated pathway, the genes that we saw go the highest, were actually downstream of TGF-beta. And this was very surprising to us because we said to ourselves, well, maybe we didn't have a knockout, maybe we had created an activating mutation, so we went back, confirmed that we did have a knockout. And what it turned out was that the genes that were being upregulated were genes in fibroblasts in those tumors. And so what we hypothesize is happening is that normally you have all these cancer cells, they're expressing the receptor, but now we've deleted the receptor, or normally sometimes it gets downregulated. You don't change the bioavailability of TGF-beta. That remains in a normal concentration. But as soon as it's out of its latent phase in the ECM, it doesn't have its receptor to bind to on the cancer cells. So without changing the concentration, we increase its bioavailability, now binds to the fibroblasts and activates them. And I think that concept of local competition between receptors is something that's very interesting for this consortium to look at, because I think there is going to be concentration dependence and gradients that we should be thinking about. So moving on from that question, this is now some unpublished data where we took that concept a step further and said, okay, we're going to take single cell data of ligand receptor pairs from ovarian tumors, and we're going to build a library to target each of these different cancer-derived factors that we predicted to be regulating macrophages in the microenvironment. And so we built a library of 35 different ProCode CRISPRs, introduced them into an ovarian cancer cell line, the ID8 model, which is a syngeneic model of ovarian cancer, had no fitness effects in vitro. So knocking out those genes did not change the cancer cells. One knockout didn't grow better than another. We then put them into a mouse, and we treated the mouse either with an isotype antibody or we give it anti-PD-1. Then we pull out and we did multiplex imaging to resolve the protein barcodes and the CRISPR. This is an example. Here we got about 6,000 different tumor lesions, but you can see this is the omentum with ovarian tumors all throughout. Then we do the staining. You can see, again, they've developed in this kind of homotypic way, so you see these very specific clones. Now, each of those clones, when we digitize them, we can then assign the particular perturbation that is in each clone. And this is an example of this is now just looking at the anti-PD-1 perturbation, what it influenced. And the gene that popped up to us was IL-4 knockout. So when we knocked out IL-4, we saw a decrease compared to the control, meaning that somehow knocking out IL-4 in the cancer cells was sensitizing them to anti-PD-1. We went and decided to confirm that, so this is now taking individual mice, injecting them with ID8 cells that have an IL-4 knockout, and you can see that this is we have a survival curve. This is a CITES development, which is a surrogate of that. And you can see that when we knock out IL-4 and give isotype antibody, there's no advantage. The mice don't survive any longer. When we give just anti-PD-1, we see very little effect. This is actually with the CITES, there's no effect. But when we knock out IL-4 from the cancer cells and give anti-PD-1, now there's a survival advantage to these animals. And the reason why we were really excited about IL-4 is this is my colleague and I, Miriam Murad, had published a paper a few years ago where we had identified an IL-4 signature in a population of dendritic cells within lung tumors. And when the postdoc, Barbara Mayer, went treated with anti-IL-4, she saw that there was a difference in this mouse model, which led our colleague Tom Moran to start a clinical trial where he took patients with lung cancer who had failed anti-PD-1 monotherapy, put them on anti-PD-1 plus Dupixent, which is anti-IL-4 receptor, and he has just a small cohort right now, but in this trial, he's actually had at least one complete responder, which is pretty amazing, just adding this anti-IL-4 antibody. Now, the thing is about IL-4, so we went back, and this is now human ovarian cancer data, and if you look, this is now looking at the IL-4 signature, comes clearly up in the macrophages in patient tumors. But when you go and look at the single cell data, you don't see IL-4 in any cell type there. And I was kind of, when I first saw this, freaking out a little bit that, okay, well, where could IL-4 be coming from? Why isn't it in the cancer cells? We started talking to Miriam about it. Turns out IL-4 and many other different cytokines don't come up in single cell data or are not easily detected because they have a very, very low abundance. And there's another reason I'll show you. So we went back, stained for IL-4 protein between the control, so they should have it, and the knockout tumors. You can see there's quite a striking difference. And it's actually quite confined. So you get these kind of regions where we think you're getting IL-4 sort of produced, and then it stops, and then that's probably why we don't pick it up easily in the single cell. Went back to the human, stained for IL-4, and again, we detected it. I'm going to quickly skip over this just to show you we confirmed with the reporter that the locus is open. And then we went and did cyclic immunofluorescence with Peter Sorger and Sandro Satagata at Harvard, where we're able to stain for 30 different markers. And what we were able to see is even though knocking out IL-4 didn't impact tumor growth without anti-PD-1, even before we give the anti-PD-1, those tumors have a decrease in macrophages, and they have an increase in exhausted T cells compared to mice that have IL-4 that are normally producing IL-4. But you can see there that at the top there are more exhausted T cells. But then when you give anti-PD-1, if there's an IL-4 knockout, what ends up happening is you get more activated T cells. You can see the granzyme and perforin positive T cells. We believe this is what's happening. So the IL-4 we think is coming from the cancer cells, polarizing the macrophages into a state which results in less of these immunosuppressive macrophages. They set up an environment where you have T cells, but they're exhausted. Then you give anti-PD-1, and now you induce the response. And I'll skip this data and just end, because I'm out of time, finishing to say we have this technology. Everyone is welcome to use it. All the procodes are on AdGene. It's a technology that allows you to do functional genomics. But I think more for this crowd to think about really this idea of the local effects of extracellular operating genes and how they dictate the microenvironment fitness. And I think the interesting thing here that I didn't really talk about is what we didn't expect is that it wouldn't be compensated for. So what's interesting to us is when you knock out something like an extracellular factor as opposed to knocking out P53 or P10 or something which operates in the cell, there's a chance for that factor to be compensated by either other cell types which could produce it, or in this case, since there's a lot of intratumoral heterogeneity, you have one clone that can, one tumor type that can still produce it beside a tumor type that can't produce it, and you would have thought that maybe that nearby tumor can produce enough of the factor to compensate for the loss of that gene knockout in the one. But it didn't happen in this case. And I think that really speaks to this idea of concentration gradients and concentration and concentration dependence that we as biologists haven't been really great at being able to resolve, definitely at the protein level. And I think it's something we have to think about how we can even figure it out from looking at gene expression structures. And with that, I'm finished. And just to thank quickly, because I know I'm out of time, the people who did the work. The ovarian cancer work was all done by Alessia Baccarini and Gurkan Malaglu, and the lung cancer work by Maxime Denan and Sam Rose, and then my collaborators, Mir Murad and Sandra Santagata and Peter Sorger. Thank you, guys.
We are running behind, but
Yes,
we have
sorry.
time for one question. Here's the one.
Hi, it looks like an amazing technology. I know nothing about cancer, but I was wondering, is it kind of like, you're talking about kind of like tissue gradients, and I'm wondering where they come from, because it looks from the fact that I don't know if I'm getting this wrong, but the fact that the tumors are not mixing between them, like the different clones are not mixing between them, indicates that there is something in the source, like in the middle of the tumor. Do you see these things?
Yeah. So it's interesting because we thought that had to do with how they seeded the tissue, but in fact, when we start to do kinetic analysis and look early on, they often don't, and you'll see like, cancer cells almost mixed, and then they start to outcompete each other. So there's something going on there where there's like a fitness advantage for one cell type over another, and sometimes it's much more local than we would have expected, and I don't think we understand that. And we see this in patient tumors all the time, this intratumoral heterogeneity, even though they're almost the same clone, and we're all looking, Fai and I were talking about that earlier, we're looking, what is the underlying structure causing that intratumoral heterogeneity? Is it genetic? Is it transcriptomic? Is it the position in the tissue? Is it because one is near a vasculature? We don't know that. These are all things we want to learn. Thank you.
Thank you so much. We're going to move to our last speaker of the session, Shin, who's going to talk about an atlas of transcribed cis-regulatory elements. Thank you.
All right, good morning. Very excited to talk to you about some of the activities that has been done at weekend in Japan and currently in Singapore. But there's a lot of overlap, and I think it'll interest many of you. I think Human Cell Atlas meetings are great because you can talk about any topic. We heard about cancer, we talked about organoids. Today you got to hear a little bit about genetics. So I think most of you already understand this concept, this intricate network of different modalities that control gene expression and gene regulation, and that's precisely what I want to talk to you about. And in a nutshell, there are cis-regulatory elements near promoters and enhancers that need to interact with each other in order to activate a gene. And if it's in the right context, they would have a smooth sailing regulatory process that can maintain a regular healthy state. Of course, if there are mutations or SNPs, either in these regulatory elements that can perturb these modules and have unwanted consequences. But what's important to understand is that these regulatory networks are very contextual to the cell types, right? So if you have here a mutation in enhancer or SNPs, but in the absence of their expression or regulatory activity, say here in blood endothelial cells, the outcome is not as severe, but if in the case of different cell type, where this activity of cis-regulatory elements are very high, in conjunction with the mutations or SNPs in enhancers, it will be disrupted, leading to unwanted consequences. So I just want to lay out this understanding that even with genetic SNPs, that it is not, of course, every cell in your body supposedly have the same similar SNPs or genetic profiles. It is really the relevance of the cellular context that will really drive this phenotype. So we want to really map these cis-regulatory elements first, and one of the strategies that we adopted was 5' strategy, and this is a lot of work done in Japan through Phantom Consortium, and this is many years ago where in profile various cell types, these are all bulk. Nonetheless, the point was that we were able to discover various promoters as well as enhancers by measuring the 5' ends of the transcript. So you can measure the transcriptional start sites, which are very cell type specific, as well as enhancer RNAs, which are types of RNAs that are derived from enhancer regulatory elements. Now we moved on to the single cell resolution and tried to capture these 5' ends using 10x genomics. And you can clearly define this 5' transcriptional start sites, as well as enhancer RNAs, which are typical bidirectional sense and anti-sense direction, which nicely overlap with known open chromatin regions. And you can really appreciate the fact that these enhancer RNAs are actually not bidirectional if you look at the single cell resolution. If you break it down to single cell, these enhancer RNAs are in fact unidirectional, but in collectively you do observe this bidirectional pattern. So here is just an example of SLAM F6 locus. At the far right, you see a very strong blue peak. That is the indication where transcription is starting. But if you look to the left in the downstream, there are small peaks that are slightly bidirectional. These are clusters of enhancer RNAs, and they overlap very nicely with ataxic signals you see in the proximal region of the promoter, as well as the distal region away from the promoter, as indicated in green. Since we have these co-activities of promoter activity gene expression and enhancer RNAs, we are able to infer their regulatory co-activities. And you can appreciate the fact that in the PTGER4 locus, upstream of that, we have a great cluster of this large number of enhancer RNAs. Activities are known to be super enhancer RNAs. And these enhancer RNA to promoter interactions are also very cell type specific, and you're able to infer these regulatory networks just based on the single cell 5' strategy. So it's a really great tool to understand both the promoter enhancer using just single assay rather than having multi-ohm assays with 10x genomics. So now that we had this tool, we then went around to various medical institutions in Japan. It was a really fun trip. We were doing road trips to different medical institutions, talking about the single cell technologies to various clinicians. And we were able to collect about 23 tissues, profile 400,000 single cells, and identified around 206,000 TCREs. So we call these transcribed cis-regulatory elements. And to make the long story short, here is a integrated UMAP, and you can also appreciate the granularity of the subtypes from the brain, and you can perform various analysis, such as differential gene expression or enrichment analysis. So ultimately, what we really wanted to do is then, can we integrate and understand the role of genetics with this TCRE atlas? And this is illustrated through this map, where you could now, for each cell type, understand the cis-regulatory element activities, but then overlap that with all the GWAS statistics, summary statistics that have been done for the past decade or two. And through various linkage disequilibrium regression analysis, we're then able to understand which cell types and which disease traits are highly associated. So there's a various computational workflow that are involved, but we've gone through various clustering, meta cell clustering, as well as generating TCRE programs and modularizing them in the context of meta cells, and running this regression analysis to understand what are the enrichment of SNPs that are associated to particular disease trait for that particular module, and we can deconvolute this at the single cell level. And just to give you what it looks like, here on the upper left, you have the conventional UMAP that are converted into metacells, and we are now able to annotate the cell types based on their modules, and then perform the linkage disequilibrium enrichment analysis for these modules, and then transback into the single cell resolution. So here's an example of what you can imagine that you're able to identify a responsible, potentially causal cell types that are associated with these disease traits. So in case of psoriasis, we have blood endothelial T cells, dendritic cells. But in rheumatoid arthritis, you have different cell types that are associated to this disease trait. Here's another beautiful example of, and the power of single cell granularity is that you have different disease traits, such as varicose veins, osteochondrosis, and triglyceride levels, but different vasculature and different endothelial cells are highly associated to these disease traits. For example, in the varicose vein, of course, the vein vascular and then the arterial are involved in the osteochondrosis, and triglycerides more so involved in capillaries. Here's another example where we can see that breaking down the brain subtypes. Up on the right, on the top, you have the GABAergic neurons, which are known to be more inhibitory. In the bottom, you have the glutaminergic neurons, which are excitatory neurons. But there's different subclusters of these neurons that are highly enriched for various disease traits that have been previously characterized through GWAS studies. So you have in the depression, schizophrenia, and bipolar have, again, different enrichment of subtypes of neurons, which are very interesting to really hypothesize which are the cell types that are responsible for exhibiting the disease phenotype in various individuals. So this is a quick summary. I just bolding the parts that I didn't have time to talk about, but there are ways that we are able to define these transcribed CREs to be more enriched in GWAS SNPs than ataxic peaks, and that these promoter enhancer activities, we can also profile their alternative usage, such as alternative promoters. And that I think ultimately previous association with SNPs to disease traits now can be filled in through single cell technology, where SNPs that are associated to promoters enhancers can then be associated to gene modules or programs, and then link them to a cell type, and then to identify the associated disease trait for that cell type. So hopefully, I think this really introduces the power of single cell genomics together with genetics that will really fill in the gaps in the linkage of responsible cell types to the disease traits. Now I'm going to switch my hat a little bit because I'm also part of the Human Cellulose Asia Network in our committee. And I think this is a great opportunity and platform to kind of highlight some of the interesting things that are happening in Asia. And really, we started out as a part of the equity efforts to how can we empower more scientists and research in Asia, and largely because Asia is home to 60% of the world's population, and there are abundant genetic, ethnic, cultural, environmental, and socioeconomic diversity. But it lacks central governance, so we really need, as a scientist, have to come together through Human Cellulose, and it was a really great opportunity to do so. So since 2017, we've been organizing meetings every year. So Piero and myself, we led the first one in Japan, but subsequently we had in Korea, Singapore, China, Australia, and Bangkok last year. And this year we're having an HCA Asia meeting in Kolkata, India, hosted by Partha. And if you guys are interested, we'll welcome you there, and it'll be in November 27th and 28th. But I just want to also highlight that through these meetings, we've been able to initiate very big consortiums such as IDA, which a Singapore team yesterday have presented, really picturing the diversity not only in genetics, but environmental aspect, as well as really big collaborative efforts that are happening in Asia. But I just want to also highlight very new consortiums that are happening around within Asia. So SCADE is a single cell autoimmune disease atlas, and they really raise big funds from South Korea, and they're able to formulate with clinicians and various research institutions around within the country. And they already generated over 10 million single cells looking at various immune cell types associated to autoimmune. And Jong-Il Kim is here actually in the audience, so if anyone want to talk to him, please go reach out, because they're going to have a very massive, important data, immune data that are associated to disease. And also just want to give a quick shout out to Jiamiao Chen, who's also here in the audience, who's also working on really data integration efforts in Singapore. So I think there's good tools that are available coming out as well. So hopefully there'll be more opportunities where some of these really big projects that are happening in Asia could also be raised to the next level in the global stage. So I'm just going to stop here. Just want to really acknowledge all the people who contributed to my first part of the presentation, where we're able to profile a large number of single cells and associated vintage disease SNPs. And also really thank Chung Chao Hon, who's really another is a partner in crime in this project, who really spearheaded this big consortium efforts together with me, and Jonathan Moody and Kono, and also Piero Canici, who's also in the audience for his contribution. So with that, I'll stop and happy to take any questions. Thanks.
In the interest of time, if there's one question, we'll take that. If not, let's have a break, and you can catch Jay during the break. One note, the breakout sessions follow this. They will start on time at 11:15, so please work out where you're going and get to the breakout session by 11:15. Thank you.